So now is the time for lightning talks. All right, so Bowen first.
Okay, so good morning, everyone. I'm Bowen. I'm a PhD student in computer science here at MIT. And in this work, we explore the idea of learning a generative model of molecular dynamics trajectories to further expand the value that can be extracted from simulation data sets. So the main idea is here at the top left. Molecular dynamics, or MD, is a very common technique used for studying various molecular phenomena, and it gives us these trajectories, which are time series of molecular conformations. Now, these conformations are generated sequentially by integrating the equations of motion. In this work, we explore the idea of learning a surrogate model of MD trajectories by learning a joint generative model over the conformation at each time step. So inspired by generative models of videos, for example, that can create a 1-minute video from scratch, we train a model to go from a trajectory of pure noise and progressively remove this noise to give us a trajectory. Now, you may ask, why do you want to do this joint modeling? We know that the underlying equations of motion are Markovian. There should be no need to learn a joint generative model like I've described. Now, typically, people learn surrogate models of MD to accelerate the simulation or the sampling. In our case, however, we argue that it is interesting to learn this joint generative model because of the new capabilities, the wider range of capabilities that it unlocks by controlling how you condition the model. So for example, if you condition the model by providing just the first frame and ask it to fill in the rest, then you get a standard forward simulator. If you, however, provide the first and the last frame, the model will try to give you a sequence of conformational states that bridges these two frames. That is a transition path. If you provide, say, for example, every 10 or 100 or 1,000 frames, the model will try to upsample your trajectory to perhaps resolve faster motions that you missed when saving your MD trajectory at 2 course of a time interval. And most ambitiously, if you mask out part of the molecule and describe what the rest of the molecule should be doing, then the model will try to in-paint or design the system to accommodate or induce those motions. Now, I want to be clear that we are far from being able to do what I've just described for large molecular systems like proteins. For one thing, the data to train such a model doesn't exist. Right? There's no YouTube of MD simulations that we can train on. But what we have done is provided a proof of concept based on training such a model for tetrapeptide systems, so very small peptides of 4 amino acids. We've created an extensive data set of tetrapeptide simulations, trained a model on a subset of those simulations, and sampled trajectories for unseen peptides. And we've validated that our model can accomplish, to varying degrees, each of the tasks that I've just described. So here on the right-hand side, on the top left, we see that our model, when used as a forward simulator, can provide quite reasonable and accurate free energy landscapes of unseen peptides. And this can be seen by comparing the MD free energy landscapes on the left and then our model's free energy landscapes on the right. Our model can also resolve, to some extent, the kinetics of the system by looking at the recovery of the relaxation time of different torsional collective variables. In the middle here, we have an example of an interpolation path between 2 macro states on the free energy landscape of a particular tetrapeptide. We do have some more quantitative assessments of this, but the explanation of those is a bit too convoluted to put on one slide. And then on the bottom right here, we see that our model, when used as an upsampling model, is able to recover the autocorrelation function of various torsional collective variables, some of which have rather non-trivial behavior at these short timescales. As for the in-painting/design, we do have some results, very preliminary results there, but not quite exciting enough to put on a highlight slide. So in summary, what we want to do in this work is just to introduce this idea and the promise of this kind of framing of generative modeling to the community, but also motivate the development of architectures that can accomplish this kind of modeling for larger systems such as proteins, and perhaps most importantly, help motivate the curation of such data sets. Now, one important caveat is that, as with any kind of generative modeling, there is the risk of hallucinations. However, unlike people who work on generative modeling in natural language, image, or video, we do actually have access to the ground truth physical model of these systems, insofar as you trust the force field that generated the simulations. So one very interesting line of work will be to see how we can detect and steer away from hallucinations by judiciously using these physical simulators in the generative process. So with that, I thank you and happy to chat more at the poster session.